Usually, when we think about our own bodies, we have this really comforting illusion of total unity.
Yeah, absolutely.
You look in the mirror and you just see one organism. Like your brain tells your hand to move and it moves. It all feels perfectly integrated, like a single seamless machine built from the ground up to be exactly what it is.
It is a very reassuring way to look at yourself. I mean, believing we are the sole authors of our own biological story. It provides a neat, tidy narrative about human anatomy.
But zooming in on the actual mechanics keeping that machine running completely shatters that illusion.
Oh, too.
Because you aren't just one organism. You're a walking, talking ecosystem. And a massive part of what keeps you alive isn't entirely human.
Which is still hard to wrap my head around sometimes.
Right. Today for our deep dives, we're looking at a remarkably dense and expansive Wikipedia article on the mitochondrion. We all know the classic 1957 Philip Segovitz catchphrase, the powerhouse of the cell.
The one thing everyone remembers from high school biology.
Exactly.
Yeah.
But looking closely at the actual biology reveals a reality that is far weirder, far more complex, and honestly a little unsettling.
Yeah, it really is.
We are going to explore their ancient alien origins,
Yeah.
their ability to make absolute life and death decisions for your cells, and the mechanics of a shocking discovery that they can actually travel outside the cell.
It's a lot to cover.
It is. Okay, let's unpack this.
Yeah.
Because before we can even begin to understand how mitochondria power you, we have to look at what they actually are.
And what they are is essentially the result of what might be the most consequential biological accident in the history of life on Earth.
An accident.
Yeah, I mean, to grasp this, we have to rewind the clock back roughly 1.7 to 2 billion years.
Okay, quite a ways back.
Just a bit. Back when life on Earth consisted solely of single-celled microbes swimming in a primordial soup. You had these ancient eukaryotic cells, which are the ancestors of the cells that make up you, me, plants, fungi, and they were relatively large but metabolically simple. Nearby, you had free-living, oxygen-breathing bacteria, likely an ancestor related to the Rickettsiales order.
Okay.
Typically, in this microscopic world, bigger cells eat smaller cells. So this ancient eukaryotic cell engulfs the smaller aerobic bacterium.
Just a completely standard microscopic Tuesday lunchtime.
Exactly. But instead of being digested and broken down for parts, the bacterium survived inside the larger cell.
Wow.
It just stayed there, and a biological truce was formed. This is the mechanism of symbiogenesis.
So it's essentially like a corporate merger where the smaller company completely refuses to integrate its IT department.
That is a great way to put it.
Or like a roommate who moved into your apartment 2 billion years ago, never left, and insists on operating entirely by their own rules.
Yes. And we have undeniable physical proof inside our cells right now that this roommate is a completely separate entity.
Because of the DNA, right?
Precisely. If you look at your nuclear DNA, the human blueprint, it's organized in those classic X-shaped chromosomes. But inside your mitochondria, they have their own completely separate DNA, and it is circular.
It's circular.
Yeah, which means it is physically identical in structure to bacterial DNA.
That is wild.
Yeah.
And the roommate insists on keeping their own appliances, too, from what the source says.
Oh, yeah.
They have their own ribosomes, which are the cellular factories that build proteins.
Right.
The rest of your cell uses what are called 80S ribosomes. But the mitochondria use smaller 70S ribosomes, which are the exact same size and structure as the ones found in free-living bacteria today.
It's a dead giveaway.
And furthermore, their inner membrane is absolutely loaded with a specific phospholipid called cardiolipin.
Right, which is a structural material almost exclusively found in bacterial membranes.
So they have their own blueprint, their own factories, their own building materials, and they even speak their own language, right? They use an alternative genetic code.
They do. In the standard genetic code used by the rest of your body, and honestly by almost all life on Earth, certain 3-letter sequences of DNA, called codons, translate to specific amino acids. Okay.
But the mitochondrial genome translates some of those codons completely differently.
Like how?
Well, in mammalian mitochondria, the codons AGA and AGG are a biological stop sign. They mean stop building this protein.
Gotcha.
But in the nucleus of that exact same cell, those same codons mean add the amino acid arginine.
Oh, wow.
Yeah, it is a fundamental mistranslation between the host and the resident.
It really is a roommate speaking a different dialect, just hoarding their own mini fridge in the corner.
What's fascinating here is how this specific symbiotic relationship was the evolutionary leap that allowed for complex multicellular life.
Because of the energy.
Exactly. Before this roommate moved in, early cells relied on simple fermentation for energy. And fermentation is terribly inefficient.
Right.
By harnessing the aerobic respiration of this engulfed bacterium, the host cell suddenly got an energy yield roughly 13 times higher.
13 times?
Yeah, that massive surplus of energy is what allowed life to grow large, to form complex tissues, and eventually to build something as intricately demanding as the human brain. So because these ancient roommates are essentially a separate entity living inside you, they play by entirely different biological rules, especially when it comes to reproduction and genetics.
They really do.
And this brings us to the mother line, because you inherit your mitochondrial DNA, your mtDNA, almost exclusively from your mother.
Right, the maternal inheritance model.
Yeah. When a sperm fertilizes an egg, it is not a 50-50 split of mitochondrial genetics. The egg is huge and packed with hundreds of thousands of mitochondria.
And the sperm.
The sperm only brings a few dozen, mostly just to power its tail for the swim.
Right. But the body goes a step further than just overwhelming the paternal mitochondria with sheer numbers. It actively hunts them down.
It hunts them.
Yeah. The father's mitochondria are specifically tagged with a protein called ubiquitin.
Oh, I've heard of that.
Ubiquitin acts as a cellular kick-me sign, marking those specific paternal mitochondria for total destruction by the embryo's cleanup machinery. It is. And because of this strict, aggressive maternal inheritance, geneticists can trace human history back thousands of generations through the female line all the way back to a single common ancestor.
Mitochondrial Eve.
Exactly.
But if we follow the logic of genetics, this maternal inheritance model sounds like a recipe for disaster.
How so?
Well, the whole evolutionary point of sexual reproduction, getting half your DNA from mom and half from dad, is recombination. It mixes things up to weed out genetic errors.
That's true.
If mitochondrial DNA is just a straight asexual clone passed from mother to child generation after generation, without that mixing, we run headfirst into Muller's ratchet.
Ah, yes.
Without recombination, an asexual lineage should theoretically accumulate harmful mutations over millions of years until the genome collapses entirely. So the mitochondria should have stopped working eons ago.
That is the big paradox. But the paradox of Muller's ratchet is solved by a ruthless, brilliant defense mechanism known as the mtDNA bottleneck.
Okay, how does that work?
This quality control process happens right at the very beginning of your embryonic development. When you are just a tiny cluster of early germ cells, those cells randomly partition their mitochondria as they divide.
Randomly.
Completely randomly. And because of that, you end up with massive variability.
Okay.
Some cells receive a high concentration of mutated, failing mitochondria, while others end up with mostly pristine, healthy ones.
So it creates a high-stakes lottery right out of the gate.
It really does. The embryo then subjects those newly divided cells to an intense vetting process using cell-level selection.
Like a stress test.
Exactly. Cells burdened with highly mutated mtDNA simply cannot produce enough ATP to keep up with the rapid demands of embryonic division.
Oh, so they just fail.
They trigger biochemical stress markers, and the embryo essentially discards them through early programmed cell death.
Wow.
Only the cells possessing the healthiest, most efficient mitochondrial lines survive to populate the tissues of the developing fetus. That's how they bypass the slow degradation of Muller's ratchet.
That makes a lot of sense. So the cell tolerates this demanding roommate because of the massive 13 times energy payoff. And it maintains pristine genetic lines through this embryonic bottleneck.
Exactly.
But to generate that kind of power, this isn't just basic fermentation anymore. It requires some heavy biological machinery.
Oh, absolutely. To understand the engine, you have to picture the physical architecture. A mitochondrion has two distinct membranes.
Okay.
The outer membrane acts like a sieve. It is covered in protein channels called porins that let small molecules flow in and out freely.
Seems simple enough.
But inside that outer shell is a fluid-filled inner membrane space, and then an inner membrane that is folded back and forth on itself over and over again.
The Christie.
Right, the Christie. And their whole purpose is to maximize surface area.
Here's where it gets really interesting. Think of that folded inner membrane like a massive hydroelectric dam.
I love this analogy.
When you eat food, your body breaks it down into simpler molecules. Inside the mitochondrial matrix, the fluid center behind the inner membrane, there's a chemical merry-go-round called the citric acid cycle.
Also known as the Krebs cycle.
Right. This cycle's main job is to strip high-energy electrons from your food. Those electrons are then fed into the electron transport chain, which is a series of protein complexes embedded directly into the inner membrane.
And as the electrons bounce down this transport chain, their energy is used to physically pump protons, hydrogen ions, from the inside of the mitochondrion across the inner membrane and into the space between the two membranes.
So you are actively pumping water uphill behind the dam,
Yeah.
creating an intense electrochemical gradient.
Exactly. You end up with a massive buildup of positive charge and physical pressure on one side of an impermeable wall.
And those protons desperately want to flow back down to balance the pressure.
They really do.
But there's only one way back through the dam, which is a highly specialized molecular turbine called ATP synthase. As the protons rush back through this channel, the ATP synthase protein complex physically spins.
It literally spins like a motor.
That mechanical spinning motion forces a free phosphate molecule onto ADP, creating ATP, adenosine triphosphate.
Which is the cellular electricity powering your heart, your brain, and your muscles. It is a masterpiece of biomechanical engineering.
It really is.
Yet biology always has a workaround. Sometimes the dam is intentionally designed to leak.
To leak? Why?
Under certain conditions, protons are allowed to slip back through the inner membrane without turning the ATP synthase turbine. They bypass the generators entirely using a specialized channel called thermogenin, also known as UCP1.
So the floodgates just open.
Basically. And when you open those floodgates, all that pent-up electrochemical potential energy isn't captured as chemical ATP. It is released purely as kinetic energy, which translates to heat.
Oh, wow.
This process, known as non-shivering thermogenesis, happens in a specific tissue called brown fat.
Which is why babies don't shiver, right?
Exactly. Evolutionarily, this proton leak is an incredible advantage. It is what keeps newborn babies warm before their nervous systems are developed enough to shiver.
That's amazing.
It is also the exact mechanism that allows hibernating animals to survive freezing winters without their blood turning to ice. The powerhouse effortlessly converts itself into a physiological space heater.
That is just incredible. But because mitochondria hold the keys to the energy grid, they also wield a terrifying amount of control over the cell itself.
They do. They are the ultimate arbiter of life and death.
Right, constantly acting as massive internal sensors. I was reading that they serve as crucial calcium buffers.
Yeah, that's a huge part of their job.
When calcium levels spike in the cell, which is the chemical signal that makes nerves fire and muscles contract, the mitochondria absorb the excess calcium to prevent cellular toxicity.
Exactly.
But if the cell becomes severely stressed, heavily irradiated, or fundamentally damaged, the mitochondria make a terminal choice. They flip the kill switch.
They release a specific protein called cytochrome C from their intermembrane space out into the main cytosol of the human cell.
And that's irreversible.
The mechanism here is absolute. Once cytochrome C hits the cytosol, it acts like a biochemical key turning in a lock. It binds to an activating factor that awakens dormant executioner enzymes called caspases.
Executioner enzymes. That sounds terrifying.
It is pretty intense. These caspases literally start snipping apart the cell's DNA and dismantling its structural proteins. It triggers apoptosis.
Which is a highly organized, programmed cell death where the cell systematically dismantles itself from the inside out.
Exactly.
So the energy provider is also the executioner.
Right.
But the real immune aha moment comes when we look at what happens if the mitochondrion doesn't neatly trigger apoptosis, but is instead violently ruptured.
Yes. This is where that 2-billion-year-old alien origin story comes back with a vengeance.
Because the rest of your body knows, on a chemical level, that the mitochondrion is essentially a bacterium hiding inside your cells.
Exactly. Normally, all those bacterial signatures, the circular DNA, the cardiolipin, the unique proteins, are safely sequestered away behind the mitochondrial double membrane.
So the immune system can't see them.
Right. But if a mitochondrion is severely damaged and forcefully spills those contents into the cell, your body's internal security system trips.
Oh, I see.
Your innate immune system relies on pattern recognition receptors, specifically ones like toll-like receptor 9. The entire job of TLR9 is to scan your intracellular environment for invading bacteria by looking for unmethylated circular DNA.
The exact kind of DNA the mitochondria have.
Exactly. So when TLR9 detects the DNA leaking from a broken mitochondrion, it doesn't process it as a broken cellular component. Because to your immune system, a ruptured organelle looks identical to a massive lethal bacterial infection.
Wow. So it responds with overwhelming force, triggering a profound inflammatory response.
It does. If we connect this to the bigger picture, Mitochondria are not just static batteries. They are the ultimate internal sensors, constantly balancing your cellular metabolism with immune defense and strict quality control.
And the moment they fail, the immune system treats them as the ancient invaders they originally were.
Exactly.
Okay, so for decades, biology textbooks pictured these mitochondria as little beings trapped inside their host cell, living and dying right where they were born.
Yeah, that was the standard model.
But a monumental discovery in 2006 flipped that assumption completely upside down. They can actually leave the cell.
Which is still mind-blowing.
The phenomenon is called mitochondrial donation, and it fundamentally rewrites how we view cellular communication and tissue repair. Researchers observed stem cells placed near dying human lung cells. The lung cells had completely lost their ability to process energy.
Right.
And the stem cells didn't just release growth factors. They actively ejected their own healthy mitochondria.
Those ejected mitochondria traveled over to the dying lung cells, were absorbed, and instantly restored the lung cell's ability to metabolize glucose and divide again.
They acted as a literal emergency battery backup.
Exactly. And the mechanics of how they travel are astonishing. They can ride inside little protective lipid bubbles called vesicles. They can float freely in the blood plasma, or the cells can actually build tunneling nanotubes.
Tunneling nanotubes are just wild to visualize.
They really are. The cells use an actin-based cytoskeleton mechanism to accomplish this. They rapidly polymerize actin filaments to push their own outer membrane outward.
Like a bridge.
Yes, extending a physical temporary bridge across the extracellular space to connect directly with a neighboring cell. They then use this microscopic tube as a highway to ship mitochondria across.
That's insane.
We see this in acute lung injuries, where stromal cells donate mitochondria to damaged alveolar tissue to keep the lung functional. Platelets can even donate mitochondria to stem cells to accelerate wound healing and the formation of new blood vessels.
But how does a dying lung cell send a chemical text message to a stem cell saying, my power is out, send batteries?
That's the big question.
And even if it broadcasts that distress signal,
Yeah.
how does the stem cell know exactly where in the three-dimensional space of the tissue to polymerize that actin filament and build that targeted nanotube bridge?
Right.
I mean, we are looking at the absolute bleeding edge of intercellular communication.
We are. The precise chemical signaling pathways, like how the distress beacons are interpreted and how the donor cells physically navigate those transport lines, remain some of the most complex, unresolved mysteries in cellular biology.
I can imagine.
But, as with all powerful biological tools, there is a dark side to this intercellular travel.
Oh.
Yeah, cancer cells, which are notoriously energy hungry, actively hijack this exact system.
Oh, no way.
Tumors create a stressed microenvironment that tricks neighboring cells. The cancer cells will reach out with their own tunneling nanotubes and literally siphon mitochondria away from surrounding healthy immune cells or endothelial cells.
Wow, they steal them.
Yep. By stealing these external powerhouses, the tumor artificially increases its own metabolic rate and builds immense resistance to chemotherapy.
It is literal biological piracy.
It really is.
Well, this brings us to a pretty harsh reality. If mitochondria are constantly working, constantly rescuing our cells, and pumping protons across membranes millions of times a second, they have to suffer tremendous wear and tear.
Oh, absolutely. Over a lifetime, the engines break down.
And this breakdown is central to the mitochondrial free radical theory of aging. Going back to our hydroelectric dam analogy, the electron transport chain, it's highly efficient, but it isn't completely leak-proof.
Right.
A small percentage of those high-energy electrons inevitably slip out of the transport chain prematurely.
And those rogue electrons react directly with oxygen in the mitochondrial matrix to create reactive oxygen species, or ROS.
Free radical.
Exactly. These are highly volatile, unstable molecules that cause massive oxidative stress. They ricochet around the inside of the mitochondrion, severely damaging its lipid membranes, its protein complexes, and most critically, its own circular DNA.
Because the mitochondria are right at ground zero of this reaction, their DNA takes a continuous beating over the decades.
Yeah.
This mechanism explains so much of our daily physical experience. That slow, inevitable drop in baseline energy as we age. It is the literal degradation of your cellular power grid.
Exactly.
It's also why antioxidants are so heavily emphasized in nutrition and longevity science. We are constantly trying to flood the system with molecules that can neutralize these rogue sparks before they shred our mitochondrial DNA.
And when your energy grid starts failing, the most energy-demanding organs in your body, primarily the brain and the heart, suffer first.
Right.
Cumulative mitochondrial dysfunction is directly linked to a huge array of conditions: heart failure, autism, chronic fatigue syndrome, and Parkinson's disease.
That makes total sense.
And certain environmental factors, like chronic pesticide exposure, can directly poison the electron transport chain, accelerating this mitochondrial damage and bringing on neurodegenerative diseases much earlier.
So it creates a devastating positive feedback loop.
Exactly. The more damaged the mitochondria become from oxidative stress, the less efficient their electron transport chain gets,
Yeah.
which causes them to leak even more reactive oxygen species, accelerating their own destruction.
But as we finally begin to map the intricacies of these ancient roommates, we are developing ways to intervene, right?
We are. A revolutionary IVF technique called mitochondrial replacement therapy, or MRT, addresses this at the source.
How does that work?
If a mother carries a severe, debilitating mitochondrial disease in her mtDNA, doctors can use the healthy mitochondria from a donor egg.
Oh, wow. So the resulting child inherits the nuclear DNA, the traits of personality, the physical features, from their parents, but they inherit a pristine mitochondrial energy grid from a donor.
Exactly. It is a profound medical advancement that directly severs the transmission of progressive, fatal energy disorders to the next generation.
So what does this all mean? We started this deep dive looking in the mirror, assuming the human body was a single unified machine.
Right.
But the reality is infinitely more complex. You are a delicate, thriving ecosystem. Your every thought, your heartbeat, your ability to fight off a virus, your body heat, it all relies on a 2-billion-year-old biological truce with an alien bacterium.
It's pretty humbling.
It really is. To truly understand human health, to cure our most stubborn diseases, and to unravel the mechanics of aging itself, we have to look past our own human DNA and understand the microscopic aliens keeping the lights on.
And the source material leaves us with one final mind-bending reality about that 2-billion-year-old truce.
What's that?
There is a single-celled organism called monocercomonoids, and even a multicellular microscopic animal, a parasite called Hennegaia salmonicola, that have evolved to completely lose their mitochondria.
Wait, really? They just lost them entirely?
Entirely. They have completely abandoned the supposedly essential powerhouse of the cell, adapting to highly specialized, oxygen-free environments by relying on alternative, deeply hidden energy pathways that we are only just beginning to comprehend.
Right.
It leaves you wondering, if life can find a way to thrive by completely evicting its ancient roommate, what other hidden energy pathways might exist in nature?
That's wild.
And millions of years from now, could human cells ever evolve to outgrow our own?